All right. Yeah, so today's talk is multivariate pattern analysis and brain decoding. Um, so in this talk, I will just start off with one quick example. Most of you probably, when they wake up early in the morning, they were dreaming of something. Even though you might not remember it, you would dream. There's always a case where you were dreaming of something. And now dreams are very intimate. It's something that you experience for yourself only, and if someone else who might be observing you from outside, they can't really tell what you're dreaming of, obviously. So now, it would of course be sometimes very interesting to know what a dream was actually looking like. And there was actually a recent study in 2013 that was conducted by the group of Kamitani, and what they were interested in is figuring out what people were dreaming of. So they had put people in the MRI, had them go to sleep, and woke them up intermittently and asked them what they were just dreaming of. And then they recorded the dream content. And then based on that, they were actually able to tell why people were dreaming, what they were dreaming of, with an accuracy of up to 75% correct. So far better than you would expect from chance. So this is a really very interesting finding, and I think it's also a very telling example for the use of multivariate decoding, because on the one hand it tells us, well, this is a very powerful method which allows you to extract information from the brain activity patterns that we have available with pretty good accuracy. But at the same time, it also tells us something about the potential implications of multivariate decoding if you want to use it in real-world applications. So this might be something that is ethically problematic if you were going to do it on someone without their consent. So now, since I'm German, I often like to start with a definition of something. So what is multivariate decoding? Or what is multivariate pattern analysis, more generally speaking? And it's actually something that is very, very difficult to define. And if you look into different review articles, they really avoid defining it. So there's a very soft definition that I'm going to choose, and not necessarily 100% correct. But the way that I would define it is that you have a combined use of multiple variables measuring the brain. That's what makes it multivariate. So you're combining multiple variables together. For example, the bold signal multiple voxels, to make predictions or to characterize different states of the brain. Okay, there's just some central questions I would like to wrap this lecture around, and one of these questions is, what are the differences between classical univariate analysis and multivariate decoding? And there are definitely a lot of differences, but that's something that's really interesting, because the way that we normally analyze fMRI data is by using classical univariate analysis. Another question is, what's actually the difference between activity and information? That will also play a role here. How does MVPA work? I mean, that's, I guess, what many of you are really interested in. And also, what can MVPA be used for? So this is like just giving you some examples where people have applied MVPA, just to see like what's actually the breadth of possible research questions you may be asking by using MVPA. So just start off with, why would you want to do multivariate pattern analysis at all? Well, I can really think of two reasons. It's very simple. One is that it often promises higher sensitivity as compared to classical univariate analysis. So just to give you one example, there's a study was conducted in 2009. They were interested in the representation of perceptual choices. And now when they ran the analysis using a classical univariate analysis, the regions encoding the choices of subjects, they found like these little blobs here that you can see here. Whereas if they ran just like a similar analysis using multivariate decoding, then you can really see that a lot more voxels here are shown to be significant. And if you have a very close look here, actually the p-value for classical univariate analysis is 0.01, whereas for multivariate decoding it's 0.0001. So you can really see that in this example, multivariate decoding was just much more sensitive. So that's one thing. In other words, people prefer methods when it's easier to get something significant, right? Okay, so that's one of the reasons why you might want to use it. Another one that's also quite interesting is that now you can actually, using multivariate decoding, you can study the representational content in a brain region rather than just the general activation in that brain region. So what does that mean, representational content? Just to give you one example, the way that people used to study visual cortex is by just looking at how strongly it activates to different conditions. So one example from the early 2000s was where people were interested in studying attentional mechanisms, and they were just interested in seeing whether activity in V1 in primary visual cortex would be modulated by attention. So that would be like a general activation level. Now, with multivariate decoding, you can actually go one step closer. You can actually ask questions about what is encoded in this brain region. And that is what I mean with representational content. What is encoded in that brain area? And we know that primary visual cortex responds quite well to different orientations that are presented to the participant. So what was done in 2005, was studied by Kamitani and Tong, is they presented participants with a set of different orientations, and they could decode what orientation a participant was shown with an accuracy of around 75% correct. And later, when they mapped the contribution of each one of these different, like the dominance of each one of these different orientations in each one of these voxels, then they would get a map that would look like this. So it just means that now, instead of just studying the general activation level, you can also look at whatever is represented in that brain region. Okay, so how does MVPA work? Again, here I'm showing you 3 principles. It's actually very, very simple if you try to understand the principles behind it rather than just understanding the implementation of different methods. But like most of these really apply for all MVPA analysis. So one is that the information across multiple voxels can now be combined. So just one example, let's say you have one voxel, and that's the way you would normally look at your activation, and you have 2 different conditions. And now you're comparing the 2, condition A and condition B, and you don't find a significant difference. So that's really a bummer. So I guess you're going to run another study. And you could look at another voxel, and again, you're not finding a significant difference between those 2. But now, potentially, if you actually appropriately combine these 2 voxels together, and this example would be voxel 1 minus voxel 2, then you might actually be able to find a difference that is also then statistically significant between these 2. So if we only knew what's the best way to combine these voxels together, we would be able to extract that information from this brain area. So this is one thing. Information across multiple voxels can be combined. Another way in which multivariate pattern analysis works is that the covariation of voxel information can be used. So what does this mean? Again, let's say we look only at one voxel at a time, and we have 2 conditions, and let's say this voxel responds, so this is like a histogram of responses across multiple trials, and it responds like this to condition A, and it responds like this in general to condition B, and you can see that they are like there's barely any difference. And in voxel 2, you might find something that looks very, very similar, so there's hardly any difference that you can see. But now, if you were actually to combine voxel activity 1 and voxel activity 2 together to create some like a 2-dimensional space of activity patterns, and now you plotted the individual trials in here, you might actually be able to find something that looks like this, where now, like originally, these distributions were strongly overlapping, but now you can actually see that you can separate them really well by just looking at the combination of these 2 and by taking the covariation of these 2 voxels into account. And a third way in which multivariate pattern analysis works is that now you can actually also uncover multidimensional representations that are encoded in distributed patterns of activity only. So what do I mean by that? Just imagine that you had only one voxel available. So this was everything you were able to measure in the brain. And now you wanted to know if this voxel is responding to all different objects that exist in the world. Well, obviously, the only way you could find that information in this one voxel is if it responded differently to every individual image. So the activation level would have to be different for all the different objects that you're showing to a participant. And that's obviously something that is very, very difficult to extract from one voxel alone. And that would only be covering one dimension, which would be just like these different objects. But just to give you another example here, let's say we have two voxels again, and we are showing a participant an image that looks just like a blue dot. And this would be the activity that we're recording for the two voxels. And let's say we're also showing them a triangle and a square. And these would be the activity, the responses of the two voxels. And then we could also be showing people large circles, large triangles, and large squares. And you don't really see much of a pattern in here. I mean, you can see that these are generally a little lower than these, but you don't really see much of a pattern here. Now, if you were to plot these two voxels against each other, you might actually find something that looks like this. And now what you can see is that embedded in this activation here is already like a two-dimensional activation pattern, so multi-dimensional representation. If you go along this direction here, you could see something that represents size. Whereas if you go along this axis here, you could see that it represents the shape of an object. So this is one way you can see that you can actually access, by using multiple voxels, you can now also access multidimensional representations. Okay, so this was how multivariate decoding works, or multivariate analysis works in general. So I could just like stop here, but obviously I want to give you a bit more information, and I'm going to get into more and more detail. So this was like a very superficial level. Now we're just diving a little bit more into it, and I just want to give you like the gist of how a typical classification analysis would be carried out, and that's also the basis for most multivariate analyses. So let's say we have a brain, and we are interested in one particular part of the brain, the lateral-occipital complex, let's say, and from that we extract what we call a pattern. A pattern just means that we have multiple recordings and we have a different number associated with each one of these. So we have, for each voxel we have a different number, and for us this would be representing the Bold signal change, for example. And now we have, in one particular example that I'm using here, we have two different conditions, and we have multiple repetitions per condition. So here we have, let's say, 8 repetitions of condition A, and here we have 8 repetitions of condition B. So now we've extracted all of these activation patterns from this brain region, and now what I'm going to do is I'm actually only going to select 2 of those voxels here. And the reason is because I can't really plot an activation pattern on this slide that has 100 voxels, because I would need to be plotting a 100-dimensional space, and that's not that easy. So just picking 2 of these voxels here, I would have the activity of voxel 1 plotted on this axis and of voxel 2 plotted on this axis here. And now for one of these examples, one of these patterns here, this might be what we get. And if I added all of them in together, this is also one way this could be looking. So this would be like for each one of these would be representing one pattern for the red and for the blue. And then the next step is what we're going to do is we're going to what's known as training a classifier. So we're going to train a classifier. And training a classifier just means that we are going to find a function that allows us to separate these dots. And then once we've trained the classifier, now these different regions, you would say they belong to the different classes. So this would be now the blue region, and this would be the red region. And now a crucial step that's now really important is you also have to see whether this prediction works on new data that your classifier hasn't seen yet. So you're going to give your classifier the data without the labels on top of it. And then you're going to make the predictions that all of these would be belonging to the blue class, and all of these would be belonging to the red class. But you actually know what the true response is, so you're going to see, okay, so I got 4 out of 5 correct for the blue, and I got 4 out of 5 correct for the red. So you're going to assign this an accuracy of 80%. And that's it. And then in the end, you would be doing some statistics to see whether that's significantly better than you would be predicting by chance. Okay, so this is just like the gist of how multivariate decoding works in general. Okay, very important distinction that really plays an important role for multivariate decoding and MEPA in general is the difference between activity and information. And I was already hinting at it earlier when I was talking about representational content, but I can't really emphasize enough the importance of this difference. So what does activity mean? Well, activity just tells us about the general involvement in a cognitive function. So again, I'm using an example here from Curtis from 2004. And one thing that people generally think is that the lateral prefrontal cortex is involved in working memory. So you find more activity in a working memory task as compared to a non-working memory task or any other control task. And by that, you would say that this brain region is involved in working memory. So this is the classical way of looking at it. Now, what this doesn't really tell you, though, is whether this brain region is just involved in working memory, so whether it might just be like important for maintaining a representation somewhere, or whether it actually is carrying the representation itself. Is it that maybe the lateral prefrontal cortex carries now a representation of whatever is to be remembered? To give you another example here, and this is now already like what fMEPA would be good for, is a study that was conducted by Christoffel and colleagues in 2012, and what they were doing is they were presenting people with these meaningless shapes, and people had to remember these. And the question they were asking with multivariate decoding was, well, can we actually find brain regions that carry a representation of these different shapes, rather than just finding brain regions that generally respond more when you're remembering something than when you don't remember something, or whenever you're holding something in working memory as compared to when you're not holding something in working memory. And they ran a multivariate decoding analysis, and what they found was that also like primary visual cortex was carrying a representation of the working memory content, something that had been shown before in 2009 by Harrison and Tong. But they also found that the parietal cortex was carrying a representation of the working memory content. Importantly, they did not find a representation in lateral prefrontal cortex. So that means that it looks as if lateral prefrontal cortex really plays more of a role in helping to maintain information that is stored in different brain areas, like here in more posterior brain regions, like the posterior parietal cortex or the visual cortex. Okay, so the reason I'm telling you this difference is because what I was telling you before is the focus on the general activation level. That is what I would refer to as activity, whereas as soon as you're interested in studying the representational content, whatever is represented, then you would be talking about the information that is represented there. And in general, you could say that these two viewpoints also have like different underlying methodological philosophies. So the classical approach, the underlying philosophy really is that the more active a brain region is, the more involved it is in the process that you're studying. Whereas the multivariate decoding viewpoint is a little different. The multivariate decoding viewpoint is the more distinct whatever you're measuring is, the more involved. So again, to give you an example, this is more like a thought experiment. Let's say you have a brain region, and this is only univariate, and the brain region in general responds this much to an apple and that much to an orange. According to the classical approach, we would say, because it's more active, it's more involved, so this means that this brain region is going to be more involved in processing apples than oranges. It's an apple region. But now what would happen if I was going to show more different fruit? So this is what would happen. It could be that for a pear, we get as much of a response as we got for the apple. For grapes, we get the same, etc., etc. So now it looks as if for any fruit that we're showing, we're finding a high response, but we don't find that for oranges. So now, according to the classical view, what people would say is, well, this brain region seems to be responding to all fruit but oranges. Whereas according to the multivariate pattern analysis approach, people might say, well, maybe this brain region carries information about oranges when you contrast it with fruit. So the conclusion that you're drawing is really very, very different depending on the philosophy that you have in mind about how things are represented in the brain. So why do we actually do multivariate decoding? So we were talking about like what is it good for, but why the focus on decoding itself. And in general, there's like two goals that you can use multivariate decoding for, and I think it's very important to distinguish these two goals. And one of these goals is if you want to make a prediction. So you want to make a prediction about states in the future. You want to find something like a biomarker. And the goal is really to maximize future correct predictions. So that means that any information that you can find in the brain is going to be useful as long as it is increasing the accuracy of your prediction. And one classical example is, let's say you want to be able to diagnose patients based on their structural brain scans. That's a classical example. People want to do this with Alzheimer's and other neurologic diseases like multiple sclerosis. So this would be an example where you really want to use the information that's present, and anything is really useful as long as it's increasing the accuracy. Another example is brain-computer interfaces. So you might want to build a brain-computer interface. And now, obviously, if this brain-computer interface wasn't working well, let's say it was only performing well in 90% or 95% of all cases, well, if you had someone using this and like riding their wheelchair on the road, then that would not be good, because if it was 95% correct in 5% of all cases, it would be making a mistake and you would have a lot of accidents. So this really has to perform extremely well. High accuracy is really, really important. Whereas it doesn't really matter where the information is coming from. If the patient is able to use their eyes and move them around or blink, it's actually fine for them as long as it works. If they can't move their body, but they can use their eyes, well, you can use a brain-computer interface and you could pick up these artifacts, but as long as it works, it's fine for that person, obviously. And another example, which I like a lot, is where also people seem to get interested in, is lie detection. So polygraphy, which is like classical EEG-based lie detection, is illegal as evidence in most countries in this world, but some people think that with fMRI you actually might be able to get a better measure of what people really, when people are really trying to be deceptive. So just imagine that one day we might have a court, and then a suspect comes in, and then they ask him a question, and he lies, and then you just see, like in the MRI, light going off. Obviously, this is a joke. A light going off, and the person's lying. So that would be counted as evidence. And well, in principle, that's possible to do something like that. I mean, apart from dragging an MRI machine into a court, I think that it's not that easy. But in principle, it's obviously possible to do this. The question is whether it's valid to do it and whether the results are actually going to be good. And based on, even though there are companies out there that promote these technologies, we're currently not at a point where we can say that we can actually really make accurate enough predictions. And it's actually questionable whether we are ever going to be able to use fMRI for making accurate predictions in the future. Okay, so just to wrap up, like we have, one of the goals was prediction. So we want to make maximally precise future predictions. Now, another goal, and that's what most of us are really interested in, is interpretation. So we actually want to know how the brain works. So the question here is more like, is there information about X, Y, and Z at all? And that is actually really interesting, and that was also what led to kind of the breakthrough of multivariate decoding and the use for functional imaging, because when people started to realize, well, the classification doesn't have to be really great, as long as it's statistically significantly better than chance, that's actually really good, because now we know that there is some sort of information about whatever we're interested in this brain region. It doesn't have to be perfect the way that we read it out, but the information is present. So it is kind of sufficient to show that there's above chance accuracy. Now, when you compare it to prediction, then obviously in prediction you can use any source of information, whereas in interpretation you really have to look out that you're using the right information sources. So you need to rule out confounds because you want to learn something about how the brain works. And just to give you another example here, it's a study that was conducted in 2005 by Haynes and Rees, and they were showing people gratings that were either presented in a visible or in an invisible state. Invisible was done by backward masking, so the stimuli were not seen by the participant. And what you can see is that In the visible case, they were performing with an accuracy around 80%, so that's pretty good. Whereas in the invisible case, if you look in V1, they could still perform with an accuracy around 60%. So that means that if this was going to be used for like a prediction, this would not be very good. But since this is statistically significantly better than chance, this means that we now can say that V1 seems to be representing invisible orientations. And that's something that's quite interesting. And you can even go to a more extreme example where here these authors are comparing, it doesn't really matter so much what they were studying, but they are really comparing accuracies that are much, much closer to chance. So they're like 52% above chance, and they're even looking at interactions here. And that is all fine as long as it's statistically significantly better than chance. Okay, so I was asking before, why are we using decoding? What's actually the counterpart of decoding is encoding. I think this is also like quite an important comparison to make. So on the one hand, we have our explaining variables. It's something that we were interested in studying. We have like a stimulus or a response or a cognitive condition. So that's like whatever we're studying. And on the other hand, we have a measured brain data. That could be the Bolt signal or EEG signal or voxel-based morphometry intensity or whatever you're interested in. And now what we normally are interested in is finding a statistical dependence between these two. Statistical dependence means there's a systematic mapping between our explaining variables and our measured data. And the typical way that we've been doing this with classical univariate analysis is encoding. So essentially what you're doing is you are going to build a model of how you think that the measured data is supposed to look like, and then you see whether your measured data is going to fit to that model. So this is like an encoding model. We are interested in predicting the measured data from our explaining variables. Whereas with decoding, the direction of the inference is the other way around. What we're doing with decoding is we're taking our measured data, and now we are trying to predict our explaining variables. Okay? But now you can actually see that both of them actually have the same goal. So if you are really interested in studying how the brain works, well, you might want to use an encoding approach or a decoding approach, but they are, in many cases, they are actually symmetrical. And what really matters here is like it's not so much whether you're using encoding or decoding itself. So it's nothing like special about using decoding when you want to study the brain function. And if you look at these different ways in studying the brain, you could say that, well, there's univariate and multivariate, and there's encoding and decoding. Because often people think that multivariate decoding is something that belongs together, but you can actually see that we have univariate encoding. This is classical like univariate analysis, or you can do like model-based, more complex approaches like what the Gallant group has been suggesting. You could be using univariate decoding. Some people have done that in the past. That would be a simple classification. You could do multivariate decoding. That's what I'm talking about mostly here. But you can also actually be doing multivariate encoding, which hardly anyone does. But there's actually methods around there for doing this, like MANOVA or cross-validated MANOVA. And then, well, similarity analysis is a method that a lot of people are using now, which I would also call a multivariate encoding approach. Although it's difficult to say whether it's multivariate or not. Okay, so this just means that we don't necessarily have to do decoding. We're just doing it because it's actually just become very popular to do so. Okay, so now coming to the questions that you can address using multivariate decoding, I would just say that's probably like 5 types of questions you can ask. I'm sure there's more, but I just want to give you five examples of questions you could be asking when you're doing multivariate decoding. One very general question is there information about your variable somewhere in the brain? It doesn't matter where it is the information present? One example here is a study that was conducted in 2010 by Plant and colleagues, and what they were asking, based on quite a small sample, that's why it wasn't wasn't like a very, very important study, but I think it was still like really, really nice. The question was, will a patient with mild cognitive impairment develop Alzheimer's disease 2.5 years later? So what they did is they had structural brain scans from people with mild cognitive impairment, and 2.5 years later they checked whether these patients had developed Alzheimer's or not. And now they were interested, like looking back in time, they took the brain scans that had been acquired 2.5 years earlier, and they wanted to see whether they can predict in the future whether someone is going to develop Alzheimer's or not. And that does have a lot of value, because if you are able to tell whether someone is going to develop Alzheimer's in the future or not, well, maybe you want to specifically focus your efforts more on helping people who are going to develop Alzheimer's, and you might want to look into why those people are more likely to develop Alzheimer's than others. And based on their sample that they had available, but again, it was quite small, they could predict around like 90% with 90% accuracy whether someone is going to develop Alzheimer's or would keep stay in the mild cognitive impairment level. And these are the voxels that were important for doing this classification. Okay, another question that a lot of people are interested in, obviously, is localization of information. One study that we conducted is which brain regions, we were asking which brain regions carry information about perceptual decision variables, irrespective of the response format. And the question itself is not so important here. So most important is we're interested in looking where this perceptual decision variable is represented. And what we did is we ran a so-called searchlight analysis, which I'm going to talk about more later, but it allows you to look where in the brain information is represented. So now we have also the ability to spatially localize the information. Another study which was quite influential was conducted in 2008, and they were studying the time course of information. So that's another thing that you could do. You could ask a question similar to that question that was asked here. At what time do you find information about free decisions of a person? So yeah, they had people carry out a task where they could freely decide to press one button or another button at any point in time they wanted. And then after they had made their decision, after they had pushed the button, they were asked, well, when was actually the time point where you carried out this decision? So the decision time point, they then aligned their responses, their fMRI data, to the decision time point when people said that that was the time point where they made a decision. And then they ran the classifier around that time point to see whether they could actually tell, before the person knew themselves, what decision they were going to make in the future. So you could, the question was, is it possible to tell whether a person is going to press the left or the right button before the person themselves actually know about the fact that they're going to press the button? So now if you look in motor cortex, what you find is that you don't really find anything before the decision, but obviously you're able to decode the button press. Whereas now if you look in frontopolar cortex and the precuneus, they found actually about 8 seconds before the participant subjectively reported to having made the decision, they could tell whether the person is going to push one or the other button. So that means that these seem to be some what they call unconscious determinants of free choices that happen before the subject actually is carrying out the response. So that's just one example to show you where studying the time course of information can actually be quite interesting. One approach that's actually one of my favorite approaches, because I think it really can tell you something new about how cognition works, is using what is known as cross-classification. So with that, you can study the association of cognitive functions. And again, to give you an example, is a study by Andre Knaps that was conducted in 2009 and was published in Science. And what they were interested in is knowing, I don't really know much about the background of this study, so I can't really tell why they were interested in it, but they hypothesized that perhaps mathematical operations like addition and subtraction could be represented in a similar format as leftward saccades or rightward saccades. So what they did is they were training a classifier on left versus right saccades. And then they tested the classifier on data that was where people were carrying out addition and subtraction. And you can see that, well, the classifier obviously, this is like prediction of rightward saccades. The classifier would predict rightward saccades if the saccade was right. And the classifier would be less likely to predict rightward saccades when the saccade is left. But now, if participants were carrying out an addition, then actually the classifier would predict that's similar to a rightward saccade, whereas there's not so much of a difference for the subtraction effect. So this indicates that, well, the only way you can really explain this result, if the classifier can generalize to this mathematical operation from only having been trained to left or right with saccades, then maybe the mental operation is sharing some representational format with the carrying out saccades. And that's why this study was really important and was published so well. And I think it's in general, it's a very, very useful approach to using cross-classification to study the association of cognitive functions, whether, for example, working memory is associated with attention or similar questions. And the last example I'm going to provide is what I call like the characterization of activation patterns. And the method that is important here is known as representational similarity analysis. So as a method has been popularized in around like 2008 by Kriegeskorte and colleagues, even though it originally has been conducted 10 years earlier by Edelman, but it didn't receive as much attention at that time. And what you can do is you can use your activation patterns that are measured for each one of, in this example, these different objects. And what you then can do is you can compare the similarity of these different activation patterns, for example, by just saying like, okay, I'm correlating these patterns to each other. And what you might end up with is a similarity matrix, or actually in this case, a dissimilarity matrix. So you can just see how similar or how dissimilar the responses are to each other. And then you can plot this using multidimensional scaling. You can plot this into, let's say, a two-dimensional space. And what you see is that all faces, human faces, are going to be very, very similar to each other. Monkey faces are going to be very similar to human faces, and they're very far away from road signs, but they also form their own cluster. Fruit and edible things are more up here, and I don't know what this bottle is doing in the middle, and this is, everyone sees that this is like a coconut, although in reality it's like the back of a head, so that seems to be a bit of an outlier. But you can see that this really nicely describes a representational space where we think that, well, this kind of makes sense that faces are represented similarly to animal faces, and animal faces are represented similarly to animals, etc. And then this really helps you to uncover something new about how the brain is working. Okay, so now, after this introduction, I will just give you like a bit more in depth, but not much more, because otherwise it will. I don't want to get into too much, like too many of the mathematical details here. I just want to give you a bit more of an idea of how classification works in general. So what I've actually studied during my PhD was looking at the representation of perceptual choices. And let's say you have your brain data, and you have two different conditions, whether the participant is making a choice to the left or a choice to the right. Now, what you can do is you can extract that these patterns of responses from from one brain area. So you have for each of these different conditions, you can extract one of those patterns here. And now, what you then do is you vectorize this pattern. So the way that you extract it, you end up with a vector that is all of these different intensities here just represent like high Bolt signal, low Bolt signal, etc., etc., for all the different voxels that you have. And now again, I'm only going to focus on two voxels here because I can't plot a high-dimensional representation here. So I'm only going to focus on two voxels. But this actually is true just for even like many more voxels that we have. And again, I'm going to plot the two against each other. So now just focus on one of these conditions here. So that's blue. And what you see is that voxel 1 has a low activity and voxel 2 has intermediate activity. So what does this mean? Well, voxel 1 is low, so that means we move up only a little bit. And for voxel 2, because it's intermediate, we're going to move up like halfway. Okay? So this point here in this two-dimensional space that we have is now representing this pattern here. Okay? So if you want to write something down, you could write down a point, or you can also say a vector in voxel space is the same as a pattern. Okay, and now for the red condition, you can see, again, like this is a high activity in voxel 1, so it's going to be moving up like this, and it's a low activity in voxel 2, so it's going to be moving over a little bit like here. Okay, and now obviously we have more than one repetition of each one of these conditions, so what we might end up with is having like multiple patterns of responses that are now plotted in this two-dimensional voxel space here. In the next step, we're going to train a classifier again. In this case, I was choosing a nonlinear classifier because it would probably be doing a really good job here, and that's it. So the other parts of how classification works, I've already explained to you. Okay, so now what we spoke about earlier is that you need to train and you need to test a classifier. And I didn't really explain to you why you have to do that. So I'm going to explain this now. So why are we training and testing classifiers? Well, again, using this example here, let's say you have your two voxels, and now I want to build a classifier that predicts like really, really well whether something is red or something is blue. And let's say, I don't know if you can think of a very good classifier that would do this, but what do you think about this classifier here? Do you think this is going to do a good job? Because I think this actually is doing an extremely good job, because you see it actually is classifying all the red ones correctly, and it's also classifying all the blue ones correctly. So we have a really, really great classifier. It looks a little odd, but it's doing a really good job, obviously, right? Okay, so now let's say I was going to apply this classifier that I've just trained here to another data set, which is measured in exactly the same way as the original data set. Well, it should in principle work really, really well, right? So it should generalize so that when I have a classifier that looks like this, everything in here should be red and everything outside should be blue. Well, I mean, I guess you already know what's going to happen. This is not going to work at all, okay? The reason why this is not working is because we were overfitting our classifier. We were choosing a really complicated classification function here on these voxels. And we can, in principle, always build a classifier that can predict all data well. There's always going to be some classifier that we can use that can predict all of our data really well. So in the goal of classification in general is that you want to find a general model that works beyond like the noise in the data. And as you can see, what I did here is I was just like using pure noise. And the way of testing this generalization is that we split up our data. We split up our data that we have into training data on the one hand and testing data on the other hand, so we can see whether our classifier is doing a good job. And this classifier obviously doesn't do a good job, but it can't because the data is just pure noise. Okay, so now if you split up your data, there's multiple ways you can do it. And what I did here is I was splitting it in half. So I said like, okay, half of my data is going to be my training data and the other half is my testing data. So that's something you could potentially do, but it's actually a very costly approach because now you're going to waste a lot of your data. You can actually be building a much, much better model if you were going to use more training data. So if in the most extreme case, you could say, well, you only leave one or two, let's say one of each of those classes out, and you put all of those data points over here, maybe the model would actually be performing a lot better because now you have almost twice as much training data available. But now the problem is that since we only have 2 data points left, our predictions are going to be very variable because either we have both of them are incorrect, or one of is correct and one is incorrect, or both of them are correct. And just by chance, obviously, this can happen, even though you might have a lot of information in your data set. Just by chance, it could be that both of them are incorrect. We have like a problem here, really. The problem is on the one hand, we want to maximize the amount of training data that we have, but also we want to maximize the amount of testing data that we have. And the good thing is that we have a potential solution to this problem, and this solution is known as cross-validation. Okay, so what is cross-validation? Well, cross-validation means you take all the data that you have available, and then what you're going to do is you're going to split it up. In this case, we're going to split it up, and we're going to just say we take 4 parts of our data, and we're going to use that as training data, and we're going to have 1 part of the data that we're going to leave out for our validation or testing of the classifier. And that will be the first iteration, and then what we're going to do is we're going to train it and test it, and we get an accuracy, and say 77%. Now we're going to repeat exactly the same thing again, but now what we're going to do is we're going to leave out a different chunk. Again, we're going to train and test our classifier, and we're going to repeat this operation. And we're going to do this over and over again until all data have served as testing data at least once. So there's also alternative ways of doing cross-validation, but this is a typical approach is known as k-fold cross-validation, in our case 5-fold. So we've actually trained 5 classifiers now. So this is our accuracy that we get for each one of these folds. Now we're just going to average that together, and this is going to be a final estimate of the accuracy. So in that way we make optimal use both of the training data and of the testing data. Okay. So just to give you an idea of like typical linear classifiers that people have been using, again, like when you're interested in studying how the brain works, people have really like limited themselves mainly to linear classifiers. And one of the reasons is that what their brain is doing is there might be some nonlinear computations that are carried out in the brain, definitely. But just imagine that you had a really complex nonlinear classifier. And now, let's say I had a really good recording from the retina. So I was able to record every individual retinal cell. And now, with my really complex nonlinear classifier, I'm going to use these retinal recordings that I have to predict the category of an object that the person was seeing. Now, if I have a really complex nonlinear classifier, like a deep convolutional neural network classifier, this classifier can do this job extremely well, even though the data has only been picked from the retinal cells. But now, obviously, I would not want to conclude from that the retina is representing objects, right? So our conclusion is, obviously, we want to know which brain area is representing objects. We don't want to just be able to read out that information. Now, this is what is known as the difference between an implicit representation. So the retina has implicitly has the information and an explicit representation. And our idea is, in general, if we limit ourselves to linear classifiers, well, we might actually underestimate the power of a certain brain region in carrying out nonlinear computations. But by using linear classifiers, at least we know that this is a computation that every brain area can, in principle, carry out. It's just pooling information together from other parts of the brain. Okay? So that's why we prefer linear classifiers. And just to give you an example of typical linear classifiers, one is the so-called Gaussian naive Bayes classifier. It's called naive because it doesn't take into account the covariance between voxels. And it's actually a very, very simple classifier. So what it would do is it would calculate the marginal probabilities of each of the classes for each voxel individually. So it's like taking this two-dimensional voxel space and projecting all of the different responses onto these axes, and you get like histograms, and these will give you probability functions. In the Gaussian case, these will be Gaussian probability functions. And now what you will do is, like, say this is your test example, you would just see, okay, what's the probability of it being red here? And what's the probability of it being blue? And then you would also look at what's the probability of it being red here, or the probability of blue. And then what you do finally is you just multiply these probabilities together, and then you get like a probability for this being red or blue, and you just pick the one that's more probable. And that's the way you carry out your classification. And this line here will just represent the equal probability plane. Very similar to Gaussian naive Bayes, but now really considering the covariance between voxels, is linear discriminant analysis. Linear discriminant analysis essentially is also using Gaussians, but now it is allowed for these Gaussians to also incorporate that covariance between voxels. But for linear discriminant analysis, the covariance is assumed to be equal for all of the classes. So the shape of this has to be similar. And if you're not interested in doing this, if you really want a separate covariance for each one of those classes, then you would be using something that's known as a quadratic quadratic discriminant analysis. And finally, just as a third example, it's one of the most popular classifiers, I would say, is the support vector machine. And the idea here is that you maximize the margin between those two classes. What does that mean? Well, essentially what the support vector machine is doing is fitting a function that somehow separates these classes, and then it's trying to maximize the border in a parallel way between these two. So you're gonna start off with a line, and you're gonna extend it in width as far as you can until you reach these different points here, and then you're gonna stop. And this is called support vector machine because these vectors here, or these points, or these patterns, these vectors are supporting the solution here. So essentially what you could also do is you could train exactly the same classifier, and removing all of these examples here and removing all of these over here, you would get exactly the same solution. So your solution is supported by these vectors here, and that's why it's called a support vector machine. And as I mentioned already, like linear classifiers are the most commonly used classifiers here. And for those interested in the mathematical background behind them, but it's very, very simple. Essentially, all linear classifiers really share the same formula. You can express all linear classifiers as a linear combination of the data times a weight. So your data, your prediction is going to be, so your class is going to be the data times a weight that's applied to them, and then it's all summed up. One other classifier I briefly want to mention is the correlation-based classifier, because it's also very popular, even though it's not like, you wouldn't necessarily say it's a linear classifier, but it's kind of close to being a linear classifier. It's very simple. What people often do is they just take the data, and let's say you have recordings for images of cats, so you presented people with images of cats, and for chairs. And what you would do is you split up your data in two halves. So you have, let's say, odd runs and you have even runs. And then you just average the data across the different runs for each of the conditions. So now you have one pattern for a cat in odd runs, and you have one pattern for a cat in even runs, and the same for chairs. Okay, so now what you would do is you would say, let's just correlate the patterns within the cats. So let's just correlate the cat patterns for odd and for even runs, and you end up with a certain correlation, and then you can also do the same for the chairs. Now, because we have recordings from the same brain, we also want to do some sort of control analysis, and the control analysis here is let's correlate between the cat and the chair, between odd and even, and we also do it the other way around. For the chair and the cat. And then our comparison finally will be, is the correlation within the category, so the reliability of a cat to a cat, is the reliability going to be higher than the reliability of a pattern between a cat and something else? And if this correlation within is higher than the correlation between, then we assume that this brain region is really carrying information about the category that we're interested in. Okay, yeah, for those people interested in a more geometric interpretation, that means that's like the smallest angular distance from a centroid classifier, but that's details. Okay, one of the last things I want to mention is there's like the different levels of MVPA analysis that you can carry out. So already hinted at it, but one way you can study information in the brain is just by looking at all voxels at the same time. And what you would get is just one value per brain. So it's not a lot, but if all you're interested in is saying whether something is represented anywhere in the brain, then that's the way you can go. And the problem with that approach, though, is that you have a lot of different voxels. You might have hundreds of thousands of voxels, and now you're having like 100 of 200,000 dimensional space. But you only have a handful of samples that you've recorded. So now finding this representation that you're actually interested in such a large space is actually very, very difficult. It's known as the curse of dimensionality. Like the more dimensions you have, the more difficult it will be for you to find whatever you're interested in. So one thing that people often do is they carry out a region of interest analysis. So you limit you limit your analysis to certain parts of the brain. And then what you would get is, again, like one value per region of interest. And there's a third approach. If you don't really have any hypothesis to start off with where in the brain something might be going on, what you can also do is carry out a so-called searchlight approach. So a searchlight approach just means that you're selecting a region of interest that's typically a spherical region of interest, and you just put that anywhere in the brain to start off with. And within that region of interest, now what you do is you calculate your classification, you're doing your training and testing and everything within that brain region, and then you end up with a classification accuracy. Now, what you're going to do is you're going to put the classification accuracy at the center of that sphere. So the center of the sphere, the accuracy is going to represent the information in the sphere. And now, once you've done that, you can now repeat this analysis for all voxels in the brain. So essentially you're carrying out, let's say, 100,000 or 200,000 classification iterations. And now what you get is you would actually get a map of information in the brain. So now you can tell where in the brain something is being represented based on the searchlight approach. Okay, I'm going to skip over this because it's not so important. Maybe I'll just end with milestones of MBPA, because I think it's quite interesting to know a bit about like the historical context, like how MBPA developed over the time, and also mention some methods that I didn't discuss today. So as I've mentioned earlier, one of the first, and probably not the first, but one of the first MEPA studies was really carried out in 1998 by Edelman. And that, as you can see, is like very, very similar to representational similarity analysis that we that we covered earlier. But this really didn't receive so much attention at that time. It was a little too early, probably. I think the most famous study for multivariate pattern analysis is really in 2001 by Jim Haxby, where he used this correlation classifier and found that ventral visual cortex can really, like, you can extract a lot of information from ventral visual cortex about individual objects. Then really the breakthrough for multivariate decoding came in 2005, where two papers came out, both in Nature Neuroscience, one by Kamitani and Tong and one by Haynes and Rees, where both of these groups showed that orientations, you could really decode what orientation a person is being presented with on the screen. And I think the reason why this really led to the breakthrough of multivariate decoding is that people now realized, wow, you can actually access a lot of information here by using multivariate decoding that you previously had no access to. So people were probably not like so convinced by the Haxby study in 2001 and like the handful of studies that were carried out between 2001 and 2005. But this really led to the breakthrough, and then everyone wanted to do multivariate decoding because that's just like very, very powerful method. In 2006, what was also very helpful was the development of the searchlight approach by Nico Kriegeskorte. Again, by Nico Kriegeskorte, like in 2008. By the way, this is research that was conducted here at NIH. In 2008, he developed representation similarity analysis, which is today also very widely used. In the same year, these model-based encoding methods have been developed, and these methods allowed using an encoding approach and then following that a decoding approach to actually predict even examples of images that the classifier had not been trained with. So the classifier could tell a category of an object even though the classifier was not familiarized with that class before. In 2011, Gontapalli and Haxby developed a method known as hyperalignment, where now we know that individual brains are very different and also functionally very different, but potentially there's a way you can functionally align them in a representational space. And what they showed is that that indeed is possible. So now what you can do is you can functionally carry out alignment, and you can make predictions actually much better between different subjects as well. And another important study was in 2014 by Chiche and colleagues, where he used representation similarity analysis and combined fMRI with MEG using this approach. And that allowed, this combination really gave them like the possibility to get like a temporarily resolved and spatially resolved signal in ventral visual areas. So they could tell when and where object information was processed, and that was quite important. And then finally, 2015 and 2016, a study by Di Carlo and colleagues came out, and they were demonstrating homologies in feed-forward architecture for like artificial neural networks in visual cortex. So essentially, this is like something that's gaining a lot of popularity right now, is using artificial neural networks and combining them together with multivariate methods of analysis. So this is like ongoing research, and it's all very exciting, but this was just like to give you an idea of what's happened in the past. Okay, so just to summarize, I started off by telling you that MVPA is more sensitive than classical univariate approaches, and that it can reveal representational content. MVPA works by combining information across voxels, so that's one thing, and by also using the covariation between voxels. With MBPA, you can investigate the informational content on top of what classic people will just study the brain activity levels. And these are two different approaches. And then importantly, we use cross-validation to reduce or avoid bias from overfitting. Okay, so if you're interested in doing multivariate decoding yourself, for those who use Python, I would recommend there's like 3 different packages out there. You could either use PyMBPA or scikit-learn for fMRI or EEG data, although PyMBPA is really focused, I think, more on fMRI. And then there's also M&E Python, which allows you to carry out these analyses for fMRI, for EEG and for MEG data. But if you're using MATLAB, I would recommend a toolbox that we have developed, which is called the Decoding Toolbox, and it's very easy to use. So it's a very shallow learning curve to start off with, and if you just want to try it out, you can just Google the Decoding Toolbox, and it runs with SPM and AFNI, has lots of different classifiers implemented. You can do searchlight analysis, region of interest, whole brain, etc. There's actually even a possibility to carry out in such an analysis just using one line of code. So that's a very, very simple way of doing it. This is just like an example. Yeah, please don't do it for your final analyses. But if you wanted to try it out, you just type this in, put in here your brick files. You need to have a separate one, though, for each run, because AFNI normally concatenates runs together. And this will be your results directory, searchlight size. And if you put this in, then it will just run. But yeah, just check it out if you're interested. Yeah, thank you very much for your attention. If you are interested in reading more, you can just look up these suggested readings here. You can also later look them up in the slides when they are available. So thank you very much. Questions?
Yeah, a question on k-fold cross-validation. Are there any, let's say you have like 100 data sets and you want to divide it into 5. Are there any rules on how you sample?
Yeah, so the question was, when you do k-fold cross-validation, how do you split up your data into different folds? What's like a good rule for doing that? So if your data is all sampled independently, then you can just split up in 5 parts, just the way that you want, or 10 parts, whatever you prefer. Machine learning people normally recommend 5 to 10, even though if you're interested in studying brain function, then you can actually split it up in more if you want. But for most fMRI data that we have available, there are dependencies in the data. And there's often like a dependency within run. So if you have multiple samples from the same run, you should keep them together, and you should not mix them up. So that's why people typically do a leave one run out cross-validation approach, and that's also what I would recommend.
Right. So, but with say independent data sets, would you want to say split them in, randomly sample them into 5 different groups, and then do that process over again to validate your results?
Yeah, so if you have independent data, whether you can then randomly sample your folds. Again, it's something I would probably recommend you to do. So I'm doing something similar for my MEG analyses, and I'm just redoing the same analysis hundreds of times because it's just very fast. And it should just give you a bit more of a precise response. But again, in most cases, our fMRI data really does have dependencies, and even if you are sampling, let's say you have a between subject analysis, so you're doing a classification between different subjects where the subjects are independent, it might actually be that there are additional variables that you need to control for. Say the gender of the subject, for example, might play a role. So that's something you might want to keep the same between the different folds. So that's just something you need to bear in mind, that there often are dependencies that you're not necessarily aware of. And if you destroy these dependencies, you might end up being okay, but it can also lead to very weird biases where you can sometimes end up getting significantly below chance decoding. Okay. Yeah?
Do you generally limit the dimensions
So let me just like follow up on that. So what do you mean with thresholding?
You threshold by some initial statistical.
Oh, okay. Yeah, I see. Okay, so yeah, so the question is, how do we select, what's a good way of selecting voxels? So there's different ways you can do it, and again, like I think it depends on what your question is. And what your question is really is like the question of feature selection, and it's a field of research in itself. And there's different ways you can do it. The typical way that most people are doing it today, even though like people have tried out all sorts of different approaches, is really by just a priori selecting certain regions of interest or excluding voxels that really don't make a lot of sense, like white matter voxels, for example. So that's like approaches that people typically use, or the searchlight approach, which is another way you can reduce your dimensionality.
The searchlight uses all voxels within the searchlight?
Exactly, it does use all voxels within a searchlight, although you could potentially also build like a searchlight approach where you say, now just also remove voxels that are white matter. I mean, there also are studies out there where people are carrying out a feature selection approach. So let's say you have a region of interest that you're interested in. Let's say it's like frontal cortex, so it's a big region of interest. Somewhere in frontal cortex, something's going on, and you're not really that interested in where it is, as long as it is in frontal cortex. Then there's many different feature selection approaches that you could carry out, but it turns out that these approaches where you threshold work actually a lot better in general. I mean, there's exceptions, but in general, such very simple approaches work a lot better than approaches that are more complex, like using nested cross-validation, where you now like trying to optimize certain parameters for selecting your voxels. For example, saying that I want to do what is known as like recursive feature elimination, where you're saying that you're repeatedly running this classification approach and you want to optimize the number of voxels that you select. And so that's something you could do in a nested cross-validation loop. And it turns out that these approaches, although sometimes they work quite well, in general they don't work that well. And I think one of the reasons is overfitting, because you don't have so much data available. Normally your data is very, very limited in fMRI, and the optimal solution that you're going to be finding there is probably not going to be the real optimal. So that's why, if you really want to do something like that, I would recommend like using very simple, what are known as filter feature selection approaches. Yeah.
How do you correct a map that you get from a searchlight?
Okay, so the question is, how do you correct a map statistically, I guess, that you get from a searchlight? That's a good question. It's a question I could give probably like a lecture on that will take an hour. But the gist of it is just there's multiple different levels at which you can look at it. But let's just say you have one searchlight map per subject, and you want to do a classical group analysis. What would, under normal circumstances, work is that you could just you normalize your data, you bring them all like into the same space, you smooth your data, you smooth your classification accuracy slightly, and you just run a classical t-test against chance. So that's what normally you would say is a good approach, and then should also work. And although some people think that you have to carry out a permutation test, it's actually not necessarily the case. But now there was a study by Ahlefeldt and colleagues, and what they showed is that the inferences that you draw from carrying out these classical random effects tests on information estimates, they're actually not necessarily correct. And the reason for that is that all information estimates in reality have to be larger than chance. So if any, the worst you can get is being at chance in reality, but you can't be any lower than chance. So what your random effects test would be reducing to is a fixed effects test. So you would be testing whether at least one of your subjects is carrying an effect, and that's not necessarily what you're interested in. So it's a very complicated topic, and it still is a matter of debate. Some people don't believe it, but I think he's correct. But for now, I would say, so what he's suggesting is that you carry out what is known as a prevalence test, so how many subjects are carrying an effect. And I think that's a nice approach, but I think there's room for improvement because this method is not very sensitive. And yeah, we just need better statistical methods. Most people still use classical tests. Okay, thank you very much.